Good evening. Greetings to one and all gathered here. It gives me an immense warmth and great pleasure to welcome you all to the Biotalk series version 2.8. Last week, our Department of Biotechnology had successfully conducted the Biotalk series version 2.7 on the topic Development of Value Added Symbiotic Enriched Feeds and Foods. Today, in our midst, we have an eminent person, Dr. Udayan Apte, who is a professor in Department of Pharmacology, Toxicology and Therapeutics, is going to speak on the topic Mechanism of Liver Injury and Regeneration. Now, I call upon our HOD, Dr. Sri Kumar, to inaugurate the event.
Yes. Good evening, my dear students and staff members. Hope everyone is doing well and safe. I'm extremely happy to inaugurate this virtual BioTalk lecture series version 2.8. As an inaugural note, I wish to share some highlights about our college and the department. St. Joseph College of Engineering is one of the top engineering colleges in India with 160th position as per the current NIRF ranking is concerned. It has got 27 years of high reputation in educational society with the NBA and NAAC A+ grade accreditations. It is having 12 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Our Department of Biotechnology was incepted in the year 2002 and having BTech and MTech programs in biotechnology specialization. Since inception, we have bagged 139 university ranks with 10 golds and 7 silver medals, and consistently we maintain a pass percentage of 100% every year. The department has got a sophisticated laboratory facilities and having research center recognition from Anna University to do PhD program. We have more than 1.2 crores, that is 12 million rupees of government funding for conducting research. This COVID outbreak caused health issues, as everyone knows, but the subsequent lockdown renders serious implications on mental health, resulting in psychological problems including frustration, stress, anxiety, lack of concentration, and depression, etc., in the society. In order to overcome these ill effects and to keep our students, teachers, and research personnel active in academics, our department has the practice of conducting series of webinars named as the BioTalk lecture series by bringing eminent personalities through webinar and sharing their research views. Today's lecture topic is going to be mechanism of liver injury and regeneration, which is a life-saving area. The liver is an interesting organ with a remarkable ability to regenerate its mass after injury. Regenerative responses involves complex and well-orchestrated phenomena occurring at the molecular, cellular, and tissue levels. In order to give more authentic explanations about liver injury and regeneration mechanism, we have Professor Udayan Apte from University of Kansas Medical Center, USA. We welcome you, Professor, and we appreciate your kindness in accepting our invitation to give a technical talk in spite of your busy schedule and time zone variation complications, etc. On behalf of the patrons, Dr. B. Babu Manohar, Chairman, Ms. Jessipriya, Managing Director, Mr. Sashi Shekhar, Director, Dr. V.S. Rao, Principal, and Dr. Parvatavardhini, Dean. I welcome the speaker, Professor Udayan Apte, and all the participants to this BioTalk lecture series. I also convey my warm felicitation to the Assistant Professor, Ms. K.R. Preeti, and her team of organizing for this webinar. All the best. Now I ask the program organizer, Mr. Alfred, to give a brief introduction about the invited speaker. Over to Mr. Alfred.
Thank you, sir. Dr. Udayan Apte was born and raised in Sangli, Maharashtra, India. After completing MSc in Zoology from Shivaji University, Kolhapur, he moved to USA and obtained PhD in Toxicology at the University of Louisiana at Monroe. He did his first postdoctoral research at Texas A&M University and later at University of Pittsburgh School of Medicine. Dr. Udayan joined the faculty of the Department of Pharmacology, Toxicology, and Therapeutics at University of Kansas Medical Center located in Kansas City, USA. Dr. Udayan's research is focused on liver regeneration, liver cancer, and adverse effects of chemicals on the liver. He has published over 80 papers, written several book chapters, and edited a book on liver regeneration. He has mentored 5 PhD students, one master's student, and several postdoctoral fellows. He also lectures PhD and medical students in the toxicology and liver biology. In his free time, Dr. Udayan is a very active in Marathi theatre in America and also in cell phone photography. Sir, it is an immense pleasure in welcoming you, and I request you to take over the session from here.
Hello, can you hear me?
Yes, sir.
Okay, should I upload my slides now?
Yeah, sir, you can do that, sir.
All right, so my camera is here, but I'm gonna look here because my presentation is here. First of all, thank you everybody. Thank you Preeti and all the organizers for inviting me and giving me a chance to share some of our work. One of your senior students, I think, Sophia is a student at our institution, and I want to thank Sophia for connecting me with your institution. It's a great pleasure to be here. So as it was introduced, I'm going to talk today about the research that goes on in my lab on the liver regeneration, mechanisms of liver regeneration, and liver injury. What I'm going to do is start with some introduction to liver, so you guys have some context of what I'm studying. So liver is the main metabolic organ in the body. It has a lot of different functions that it does. It's a unique organ because it does functions that nobody else can perform, and so it is very critical for maintaining function in the body and life. These are the functions that I've listed. There are others, but liver is the main function, which liver is the main organ where we make bile, which is very important for fat digestion, as well as the bile acids are also signaling molecules. Liver is the main site of drug metabolism or detoxification, really, because liver will see everything we ingest, and thus gets exposed to a lot of toxic chemicals, anything that's basically in your diet, and liver's function is to detoxify that. It's a prominent organ that does most of the micronutrient metabolism. So it absorbs a lot of these chemicals, absorbs a lot of the macronutrients, fats, proteins, glucose, everything from our food, and actually converts it into things like glycogen, and then also involved in fat metabolism and protein synthesis. It's the organ where urea is produced, so it is very important for getting rid of nitrogenous waste. One of the problems with failing liver is that when liver fails, these nitrogenous wastes in the body start to build up, and they have been implicated in what's called as hepatic encephalopathy. There are other functions of the liver. A very unique one is liver is making a lot of different proteins, and one of them is making coagulation factors. So liver plays a very important role in blood coagulation, and therefore if your liver is not functioning well, your blood coagulation function goes down, and the patients of liver failure are at a very high risk of bleeding. And so that's another very important thing that liver does. Now, this is just a general idea of how liver looks and where it is. Liver is present right below our diaphragm. It is very well protected by a rich cage, and it is connected with the gastrointestinal system using blood supply. This is how the liver looks. This is the human liver. It has four lobes. Left lobe, right lobe, which is the biggest part of the liver. And if you flip it from this other side, it has a caudate lobe and a quadrate lobe. Right in the middle is this area called as falciform ligament, where all the vessels will enter the liver and exit the liver, like gallbladder, for example. We use mouse models to study liver, and so this is a picture of a mouse liver. It has pretty much the same lobes, but they look slightly different. They have the median lobe, the left lobe, caudate lobe, etc. And it's more lobulated than a human liver, which looks more like a big chunk of tissue. One of the most unique things about liver is its blood supply. Now, as you probably already know, all organs in the body have sort of two blood connections. One is where blood comes from the heart. Fresh oxygenated blood comes from the heart, and it is supplied oxygen and nutrients to the tissue, and that's the arterial blood supply. And then there is a venous blood supply that takes away all the deoxygenated, nutrient-less blood away back to the lung and heart, and then to the lung. Liver, of course, has those two blood supplies, but in addition to that, liver has this unique blood supply that's called as a hepatic portal vein. So liver will get a blood supply directly from the heart. This is all the fresh bloods coming down here. But what happens is the aorta is also supplying blood to all the gastrointestinal tract, all the way from esophagus down to colon. And the blood supplied, the blood from these organs, after it is collected, all the nutrients that are obtained from the digestion process are actually stopping in the liver before they go to the heart. And this happens because all the blood supply from here actually is collected into what's called as a hepatic portal vein. And this hepatic portal vein will bring that blood, which is deoxygenated because oxygen is used up by these organs, but it is super nutrient rich. It is the most nutrient rich blood in the body because it is collecting all those nutrients. Anything that you eat, and I know it's nighttime in India, all that really nice dinner you had, all of that stuff is right now going to your liver through this digestion process through the hepatic portal vein. And then the deoxygenated blood, of course, drains back to the heart. So this unique blood supply really makes a very important, it's very critical for the function of the liver. As you can see, not only nutrients, but all the types of medicines we take through our mouth, or anything that we get exposed to through our mouth, will actually end up in the liver. And because of that, liver actually gets exposed to a lot of toxic chemicals on daily basis. Of course, there are drugs that we take, alcohol, viruses, all the environmental pollution, anything that goes through your mouth is going to end up in the liver. And it's liver's job to detoxify them. And so liver is constantly getting injured. However, even with that, liver is not failing all the time because it has this remarkable capacity to regenerate. So ability of the liver to regenerate is one of the most fascinating things about liver, not only about liver, but about our body. And it has been known for a long period of time. And, people like us who study liver regeneration for a living like to believe that ancient Greek people knew about liver regeneration. So there's this myth of Prometheus. You probably know that Prometheus was a titan, and he stole fire from the gods and gave it to people, thinking that we'll make a good use of it. Now, what we did with the fire is a different question, but all the gods were very angry. They said, look, this cannot be tolerated. So they went to Zeus, the king of gods, and said, you have to punish Prometheus, make an example of him. And so Prometheus condemned, Zeus condemned Prometheus to what's called as eternal torture by tying him to the Mount Atlas, rock on a Mount Atlas, and opening up his belly and asked the eagle to eat his liver. And so the eagle would eat Prometheus's liver and it would regenerate in the night. And then the eagle would come back and eat the liver again. And thus he was condemned for eternal torture. And this mythological story has been a staple of painters throughout last 2000 years or something. And you can see this is one of the paintings that I'm showing, which is in the Vatican City Museum, where Prometheus is bound, the Zeus condemning him, and the eagles eating the liver. Now, through the ages, people have kind of understood that liver regenerates, but the first well-documented study of liver regeneration was performed by two pathologists in Chicago, Higgins and Anderson. And they published this report in 1931, where they used a rat, and what they did was they took out about 60 to 70% of the liver using a surgical method. And they let the remaining liver, whatever 30% liver was left, to regenerate back. And they reported that within about 2 weeks, the liver regenerated back and literally got all its mass back. And so now that is very important, because that really forms the basis of a lot of treatment in the clinic. People who have tumors get something called as hepatectomy, where the part of the liver is cut down where the tumor is, and the remaining liver will grow. Also, other things happen, when people are getting living donor liver transplants. In liver transplant field, there are two types of transplants. One is where somebody's died and you get their liver and donate, that's put in the recipient. But nowadays they also do what is called as living donor liver transplant. A person next to kin who is ready to donate, healthy person, donates their liver for the diseased person, and liver will regenerate in the donor and the recipient as well. And so, we can do all of this because liver has this ability to regenerate. Now, liver's ability to regenerate is also very important when it comes to toxicant-induced liver damage or chemical-induced liver damage. A lot of medicines, when there is an overdose or environmental chemicals, they can cause liver injury, but there is actually very nice regenerative response. And when we're talking about regeneration, we're talking about cell proliferation. I'm going to get to that here in a second, but liver can restore its function. And so these are a lot of different things that are very much important for regeneration. And over the years, the liver regeneration has been really well studied. You can see the number of papers that are published each year in this area. It is high. Even after this, there is still a lot that we don't know about it. So when we're thinking about liver regeneration, you have to think that the crux of regeneration, the crux of liver regeneration is cell proliferation. At the center of it is the fact that when cells in the liver die, the remaining cells in the liver will divide and actually add new cells. There's another way of thinking about it, right? The remaining cells in the liver could increase in size, and that would be called hypertrophy. And when cells will actually divide and add new cells, that is called hyperplasia. So liver regeneration is conducted by hyperplasia mechanisms. And so out of the lot of different types of cells, in the first cells that divide are hepatocytes, the main cells of the liver, followed by other cell types in the liver. And this is what happens in rodents, in rats and mice. Within about 10 days, you complete that process. And this process is governed by a lot of different redundant signals. You have to realize that liver regeneration is so critical to maintain life that there are lots of different mechanisms that are redundant and that are put in place. So if one mechanism fails, something else can take over. At the center of it is cell proliferation, that is regulated by delayed early and immediate early genes, things like c-Myc and c-Fos and all those. And these are regulated by a lot of different kinds of extracellular signals, cytokines, growth factors, bile acids, hormones, things like that. And on the top of that, there are these larger regulatory mechanisms, right? For example, the circulatory mechanisms, circadian rhythm plays a role, metabolic signals play a role. And so this is a very interesting process, and this is basically what we study. So in my lab, we have been studying liver cell proliferation. So when I introduce my lab generally to people, I say I study normal and abnormal proliferation in the liver, and I study that in the context of either livers regenerating after surgical or chemical injury, after environmental exposure, during liver development, and an extension of all of this is liver cancer, because in liver cancer you're going to have cell proliferation. But unlike all these three different ways, it is not, especially these two different ways, in these two ways, especially in liver cancer, liver proliferation is uncontrolled. In regeneration, you're controlling liver cell proliferation using lots of different mechanisms. So it starts at a point and shuts down at a point. In case of cancer, that doesn't happen. And so today I'm going to share with you a story on what's called as acute liver failure and how liver regeneration plays a role. So this sort of pictorial shows you different causes of a clinical condition called as acute liver failure. Now this is defined as a disease in the liver where a person's liver was normal, there was absolutely no injury, and suddenly something happens and they acutely get sick because their liver starts failing, and they get sick within a day or two, and the outcome is either they will regenerate the liver or they will have a liver failure and they die. In some cases, fortunate individuals will get a liver transplant as well. So if you start looking at different reasons why acute liver failure occurs, you will see that acetaminophen overdose is a major cause in the Western world. Not so much in India, where we have anti-tubercular drug injury in India quite common, as well as hepatitis E virus. But in the Western world, overdose of acetaminophen is a major cause. So what is acetaminophen? Well, you guys know it as paracetamol, right? Paracetamol is one of the most common medicines on the market. It is actually a very safe drug. I don't want you to start worrying, should I take paracetamol or not? Remember that paracetamol, when you take at the right dose, is a great medicine. I am a toxicologist by training, and the basic rule of toxicology is that dose determines the poison. Basically, we think that anything is a poison at a certain dose, and so all our medications can be very toxic, if you overdose them, and that's certainly true for paracetamol. So it is a very widely used medication, and we know that in the Western world, these are data from United States, and we know that about 50+% of all the acute liver failure cases are due to overdose of paracetamol. Some of those are intentional, some of those are unintentional, and we'll get to that later in discussion if you want to. We also know, fortunately, about 60% of the paracetamol overdose patients, we call it acetaminophen here in the Western world, mainly, and it's abbreviated as APAP. So over 65% of these patients will survive, and 30% patients will not survive. And so how does paracetamol or acetaminophen cause injury? And this has been well studied over the last almost 30 years, and we're still discovering new things. So this is the structure of acetaminophen. Normally, acetaminophen is detoxified by glucuronidation and sulfation. One of the functions of the liver is to get rid of any xenobiotic, any foreign chemical in the body, and they do things like conjugating it to more water-soluble things and getting rid of them. Now, very little acetaminophen normally when we take it is available for an enzyme called as CYP2E1, which can convert it into a very reactive molecule called as NAPQI. NAPQI is highly reactive, it can cause lots of tissue damage. Fortunately, we have a way to prevent that. There is an antioxidant molecule called as glutathione. It is actually a tripeptide. It is made of 3 amino acids, and glutathione can conjugate with NAPQI and several other oxidative molecules and detoxify them. Now, during overdose, what happens is all these detoxification pathways are overwhelmed. You use up all the glucuronidation, sulfation, and glutathione quickly, and then what happens is all you got is a lot of NAPQI in the liver that is ready to cause a lot of damage. And then this will cause a lot of damage. Now, the therapy that is currently available, there's only one therapy for acetaminophen overdose, and that is N-acetylcysteine. You can infuse IV infusion of N-acetylcysteine is given to the patient. And what it does is tries to produce more and more glutathione. It's a slow process and it's not very effective. Now, all this free NAPQI will cause tremendous amount of cellular necrosis by a cascade of events. It forms protein adducts, causes extensive reactive oxygen species, oxidative stress, and that turns on a cascade of signaling molecules leading essentially to a mitochondrial permeability transition and mitochondrial burst. And then cells die, and once the cells die, cell-independent mechanisms can take over. Inflammation can start, and there is also a release of cell death proteins. Now, fortunately, liver has this ability to regenerate. It's not just going to sit there and watch the damage. And studies from our group and a lot of other groups have shown that liver regeneration is very critical, because if you have regeneration, you will recover. But patients who do not have regeneration, they will get acute liver failure. In fact, we think that you have those 65% patients who survive, they actually have better regeneration as compared to those who don't survive. And this essentially is kind of divided into, these are the sort of the three phases of acetaminophen toxicity. And so we come up with this three-stage model of acetaminophen induced acute liver failure. First is the initiation model, where you have starting of cell death. Then there is a progression model where there is a lot of inflammation, the injury spreads beyond the initial cell death, and thirdly you have a recovery and regeneration phase. And my research is mainly focused on this recovery and regeneration phase. Now, I'm going to show you these data to tell you that regeneration is absolutely critical for survival in this case. How do we know that? Well, there is a paper here, and there are others as well, but these are the data published several years ago by a Swedish group, and they showed that what they basically did is to measure alpha-fetoprotein in serum. Now, alpha-fetoprotein is a marker of proliferating hepatocytes. Whenever liver cells are proliferating, they make this protein. It comes in the serum and you can measure it. And so they looked at a lot of acetaminophen overdose patients, more than about 220, and they found that patient who survived had significant increase in alpha-fetoprotein, indicating that there was proliferation. Patients that died, they had this flat curve where they did not make AFP, indicating their cell proliferation was lower. So these are excellent data to understand that regeneration is critical for survival. So our idea was to kind of go back and see that we wanted to model these two groups, right? You have patients that survive, and then you have patients that die. And how do you model that in animals? Okay, so that's where my background in toxicology was very helpful. My PhD mentor, Dr. Hari Mehendale, had spent about 20 years of his life, research life, coming up with this concept that as the dose increases, the ability of animals to regenerate also goes up. However, this ability stops at a certain place. And so I'm going to use this schematic to explain that. So here you're seeing increasing dose here, and injury and regeneration. And as the injury increases, we also get increased regeneration. However, beyond a particular dose of a chemical, you don't have regeneration. The ability of the tissue to regenerate is essentially blocked. And we don't really know the reasons. We are now understanding, as I'll show you here in a minute. And when that happens, beyond that dose, when people are exposed to doses higher than that, injury will simply progress and die. And Dr. Mehendale showed this using a lot of different chemicals. And we said we will use acetaminophen to test this hypothesis. And so what we did was gave animals a dose of 300 milligrams per kilogram, which is below the threshold. So all these animals, they will get sick, but they will recover. and then we gave other group of animals a dose of 600 milligrams per kilogram. They are going to get sick too, but they're not going to recover. Some of them are going to die, others are going to take much longer to recover. And so this is what we call the incremental dose model. Okay, and so we are we have been studying this. I'm going to shut this down. Sorry, the sun is right in my face. We have been using this model to kind of as a starting point to understand how liver regenerates. So we got this very interesting incremental dose model, and these are some of the data that we published many years ago. What you're looking here on the top is the H&E sections of mice given 300 milligrams per kilogram dose or 600 milligrams per kilogram dose, and followed over a period of 4 days. And you can see that in the 300 milligram per kilogram dose, you get injury that slowly reduces. Patches of injury are lined here by yellow dotted line. But in 600 dose, it takes much longer to recover. You can see that in serum markers, ALT, as well as a percent necrosis, that the lower 300 milligram per kilogram dose, which we call as a regenerating dose, there is injury, but they recover. And 600 dose, there is higher injury, and they do not recover. And so we have this model where we have two groups that have now different injury. Let's look at what happens in regeneration. This is a marker of cell proliferation, which is a way we measure regeneration, looking at pCNA here. And we have looked at, we have counted cells that are proliferating here. And this is a graph that can help you explain. You can see that in the 300 milligram per kilogram dose, regenerating dose, there is massive increase in cell proliferation. In 600 dose, there is an increase, but it is quite delayed, almost 48 hours delayed, and it is much lower. And so these data also suggest that, we know that from these data that having the proper regeneration that start at the right time is critical for survival. So we followed this further and looked at molecular mechanisms of regeneration. So what at the crux of regeneration, as I said, is cell proliferation, right? One of the molecules very critical for cell proliferation is cyclin D1. It is a protein that pushes cells, quiescent cells, into cell cycle. And you can see in 300 dose, we get massive activation of either mRNA and protein of cyclin D1, and not anything going on in 600 dose. And so it's true about a lot of these other cell proliferation markers. We also looked at P21 mRNA. Now, P21 is a molecule that inhibits cell cycle, right? And so we found that there is massive induction of P21. It's 600 dose and not so much in the lower doses. And so we are very, that these data suggested that there are two different things that are happening. One, that there is proliferation going on in the lower doses, regeneration is turned on, and at higher doses, there is probably an active inhibition of cell proliferation that is going on. Now, your question might be that, hey, you gave a double dose to these other mice. Maybe there is so much damage that there is no liver left to regenerate, and that is actually possible in regeneration field, where what we say that there has to be critical mass left in the liver to regenerate. Okay, because what is happening is when liver is regenerating, it is still doing its function. So not everything is shut down, and so that is very important to know. So we looked at that. We stained the 600 dose mouse liver sections with a molecule for a molecule called HNF4 alpha, which we study in the lab. And only functional, healthy hepatocytes have HNF4 alpha. And that basically showed us that although about 35% of cells are dead, the remaining 70% liver is still there. It's not that there is loss of critical mass. So the cells are not dead, but they cannot regenerate. And I think about them more like what I call as PTSD hepatocytes. They're shell-shocked, there's massive injury and they don't know how to respond to that. And there are mechanisms that are just stuck and won't move forward. And so we have used this model to study a lot of different molecules, and we have published over 15 papers now in the last 10 years to to identify role of different molecules. We are currently doing work on these. These are all published. We are still working on some of these too. A couple of these paper on HNF4 alpha and YAP are almost ready to be submitted. So in the remaining time, what I'm going to do is just to show you one study that is pretty interesting, and that's on EGF receptor. Now, you guys are probably familiar with this receptor called epidermal growth factor receptor, and EGFR signaling. EGF receptor is a very important molecule. It has many ligands, EGF, TGF alpha, amphiregulin, and so on. And these are direct mitogens for hepatocytes. Our interest in EGFR started because we know that all these molecules can trigger cell proliferation in hepatocytes. They're important for liver regeneration. And so we were very interested in knowing what's going on with this in our model. And we also know that the molecules play a role in general in cancer and regeneration. So we started looking at EGFR, and we were very surprised. What happened is, this is Western blot analysis. This is total EGFR. You're looking at 300 dose data here and 600 dose data here over a time course of 0 to 96 hours. And you can see there is total EGFR in both groups, and phospho-EGFR, which is the active form of EGFR, is present also in both groups. Not only in both groups, you can see there is a dose-dependent higher activation of EGFR. So we were very surprised. We were assuming that these mice here don't regenerate, and so a molecule that is involved in regeneration will be lower expressed. But it's not like that. You can see that it is highly expressed, and in fact, we saw activation of EGFR as early as half an hour after acetaminophen overdose. Now remember, over the I showed you that there is a time course of things that happen, right? You have initiation, then progression, and then recovery. So mechanisms involved with regeneration don't really start or don't really show themselves until about 24 hours after acetaminophen overdose. So we did not expect to see any EGFR activation within the first 24 hours. You're seeing it within the first half an hour. So we were wondering what's going on here. Maybe there is something that EGFR does in injury that we don't know of. And so we looked at primary human hepatocytes. We are fortunate to have a resource where our transplant surgeons give us a little bit of the liver that is going to waste when they do transplant. And we have technicians who make fresh human hepatocytes, and then we can use them for research. And in culture, you cannot simulate regeneration. That's another interesting thing. Liver has this ability to regenerate, but hepatocytes, as soon as you take out from the animal and culture them, they lose that ability. But you can still study the mechanisms of injury, the first part. And we looked at that, and here also we found very early activation of EGFR. So the question was, is EGFR involved in injury or regeneration? So we have a tool to study that. There is a chemical called as kennertinib, and kennertinib is a very potent and irreversible EGFR inhibitor. And here is an example of the preliminary data from those studies, where we took mice and gave them acetaminophen, and another group got acetaminophen and they got EGFR inhibitor, and we measured activation of EGFR, and we saw a significant complete block of EGFR activation. So we knew that this inhibitor works. So we designed a very simple experiment. We gave mice acetaminophen. We waited for 1 hour for all those things that produce injury to happen. The conversion of acetaminophen to NAPQI and starting of cell death. And then we gave them the EGFR inhibitor, and then we studied animals over a time course. And here is what we found. We found that early inhibition of EGFR almost completely prevented injury. The EGFR receptor blocker, just completely blocked liver injury. There is no necrosis, no ALT, and you can see that here in the cell, H&E as well. And so when we started wondering, that basically told us that EGFR activation that we saw early on actually plays a role in liver injury. We actually wanted to study its role in regeneration, but we were surprised that it's playing a role in injury. So now, of course, we wanted to know what part of the injury mechanism the role EGFR is playing a role. So this is a schematic, essentially a shorter version of the big schematic that I showed you. Acetaminophen is converted to NAPQI. There's glutathione depletion, protein binding leading to mitochondrial injury and cell death. So we decided to look at each of these phases. First, we looked at glutathione depletion and protein binding, and we didn't find any difference. There is no difference in formation, depletion of glutathione or formation of adducts between normal animals given, between acetaminophen alone animals and animals giving acetaminophen followed by EGFR inhibitor. The next thing we studied was JNK activation. There are a lot of studies that show that a kinase called Janus-associated kinase or JNK kinase is very critical for acetaminophen overdose induced liver injury. If you block JNK activation, you can actually prevent injury. And so we looked at that, and again, we didn't find any difference. Here there is 0 hour, 3 hour, 6 hour, and 12 hour, either acetaminophen alone or acetaminophen plus EGFR inhibitor. And you can see there is really no difference between these groups, not only in the total cells, but also in mitochondrial fraction, because JNK activation can happen in cytoplasm as well as inside the mitochondria. So you have to check both, and there was no difference. Then we went on to look at mitochondrial function. We have this instrument called Seahorse, and we've said, how about we look at what's going on with the mitochondrial function? And lo and behold, we found that mitochondrial function was improving when we gave EGFR inhibitor. You give acetaminophen alone, this is your saline control, no treatment. You give acetaminophen, there is a decrease in mitochondrial oxygen consumption rate, and then that is restored back when you gave EGFR inhibitor. So we were very surprised to see that. And the question became, well, is actually there EGFR in the mitochondria? Now you remember, this is a cell surface receptor, but there are a lot of data suggesting that it is actually internalized and it does things. So we wondered whether that's happening after acetaminophen, and yes, it does. So here are the data to show that. This is total cell lysate, and this is the mitochondrial fraction. 0 hour and 1.5 hour after acetaminophen, you can say there is increase in mitochondrial, there is increase in EGFR activation. Now, if you look at the mitochondria at 0 hours without any acetaminophen treatment, there is no EGFR, but it increases within 1 hour, 1.5 hour after acetaminophen. If you give an inhibitor, it doesn't. And so now we know that EGFR is actually internalized and goes to mitochondria. We don't know how that happens and what's going on there, but these are some very novel findings we have. And this EGFR activation in the mitochondria is very important, because the next thing that happens in acetaminophen overdose is mitochondria burst, and they release all these proteins called as AIF and endonuclease G. We found that the release of these chemicals is prevented when you give EGFR inhibitor. So this is looking at AIF and SMAC, and you can see that there is significantly less AIF in the cytoplasm coming out from mitochondria when you give EGFR inhibitor. So EGFR goes to mitochondria and does something which actually promotes mitochondrial damage. And if you block EGFR activation, this process is not happening. So, whenever you have something new, you want to test it against the current therapy, right? The current therapy is giving N-acetylcysteine, as I told you initially. So we said, look, we could block EGFR and we can prevent injury. So this could be a very nice new therapeutic. How about we compare that with the new, the current therapy? And so we did that. Here, one of the things with N-acetylcysteine is that it will work in patients only in the first 24 hours. In mice, which we model those patients in, it works in only about first 3 hours, 2 or 3 hours. Beyond 4 hours, it doesn't really work well. And the reason is all those mechanisms of converting acetaminophen to NAPQI take place within the first 2 or 3 hours in the mice, or in the first 24 hours in people. And after that, those mechanisms are not going on. Things have moved forward. So if you block, you have to block them, you have to block them in that time frame. And so here is an experiment where we took acetaminophen mice and gave them acetaminophen, or 4 hours later we give them NAC, or we gave them EGFR inhibitor, or gave them both acetaminophen, NAC and EGFR after acetaminophen. And we found that the only thing that can prevent injury or reduce injury substantially was the combination of NAC and EGFR. And so we were very happy to see that there is something going on here. However, we were still wondering, what if we give EGFR inhibitor late? And there is a reason for that. What happens with these patients is that, you can prevent acetaminophen injury by blocking junk, blocking other things during that first few hours of time. Okay, in people that's about 12 to 24 hours. Unfortunately, majority of the patients will come to ER or to the medical system, to the hospital, after those things have happened. They take overdose, or they're getting overdose without knowing, and they don't get sick right away. It takes about 6 hours to 8 hours to feel bad, and then things get really bad after 12 hours, right? And then they're coming to the hospital. By the time they're in the hospital, all those mechanisms that I showed you in the initiation phase, they've already happened. You cannot block them anymore. The only thing you can really do in the hospital is to actually, try to promote regeneration. And so we are always trying to find out whether we can use some sort of delayed treatment in the hospital. So we said, why not we try giving EGFR inhibitor late and see what is going on with it? What is EGFR blocking still prevents injury if we get late enough? And so we did that experiment, right? We took mice and gave them acetaminophen, and we waited for 12 hours now. The initial experiments were done at 1.5 hours. Now we waited for 12 hours and then gave EGFR inhibitor, and then looked at 24 and 48 hours. And this time we were in for a major surprise. When we blocked EGFR very late in the game, we found that injury actually increased. Here is histology and ALT. You can see at 24 hours there is no difference between injury between acetaminophen and acetaminophen plus EGFR. But at 48 hours, these mice have started to recover, and the EGFR inhibitor mice are actually getting worse. Not only they get worse, they actually also start dying. When we looked at regeneration, we found that they have substantially lower cell proliferation. So delayed inhibition of EGFR actually prevented regeneration. This was the experiment we should have done first, actually, because we know that EGFR plays a role in regeneration. Now, when you block it early on, it prevents injury. When you block it late, it prevents regeneration. So it's playing sort of a dual role in this process, is what our data suggested. This is just looking at molecular mechanism, cyclin D1 and all the other things that drive cell proliferation. And you can see at 24 and 48 hours, if you give EGFR inhibitor, all the molecules that drive regeneration are downregulated. And so early on, yes, EGFR might be beneficial. Preventing EGFR activation might be beneficial, but a delayed prevention is going to be a problem. And so to summarize this story, we found that a rapid EGFR activation, maybe due to glutathione downregulation or depletion, is driving injury, but EGFR still plays an important role in driving regeneration at later time points. And so we now know that it has this pleiotropic role, playing a role in both injury and regeneration mechanisms. So I'm going to end here, with the summary that liver has this remarkable capacity to regenerate. It is critical for survival, and it is driven by tremendous tremendously complex and redundant signaling mechanisms. And in the end, I want to thank all my lab members, former lab members and current. Bharat Bhushan is my student who did most of the work. I showed you he is now a faculty member at University of Pittsburgh. My mentor as well as my collaborator, Hartmut Yashki, is very critical for these studies. He's an expert in world expert in acetaminophen overdose studies, all my funding. And I want to end with a picture of a lake near our house here. One of my hobbies is to do cell phone photography, and the picture that I took a few days ago to just give you how Kansas looks like. And I'll be happy to take any questions. Thank you.
Thank you, sir, for this wonderful lecture and enlightening us on the topic mechanism of liver injury and regeneration. Sir, we have few questions. And the first question is, what is the percentage of survival after liver transplant?
After liver transplant, if it is done correctly, I think a lot of people, most of the people survive. It actually depends on how sick the patient was before the transplant, but in majority of the cases, the survival is very good. It does depend on a lot of different things. What the disease, what kind of disease the patient had, what kind of post-operative care the patients receive, and there's a very interesting paper that came out a couple years ago. They looked at people who got liver transplant following acute liver failure. The reason of acute liver failure might be different. They had different reasons why they got acute liver failure, not only acetaminophen, but other things as well. I'm going to stop sharing so I can see you guys better. They found out that people who come from lower socioeconomic classes had poor outcomes. And there is really no reason. They were not able to figure out what exactly happened, but the general idea is that there might be that might be happening because of not having better health care access to these people, which is very important. Post-transplant life is not easy. These patients are generally immunosuppressed for rest of their life. Because you don't want the liver to be rejected by the body, so you have to suppress the immune system. Now, when you suppress the immune system, of course, you are inviting a lot of diseases, right? Infectious diseases and so on. And so that's where things can get very tricky.
Sir, and the next question is, does COVID post-treated condition has some impacts on the liver?
There are data on that. Yes, COVID, induced liver injury is now being reported quite well in the literature. Liver failure is one of the signs of, or one of the sequelae of COVID infection, a severe COVID infection. Unfortunately, COVID, the virus can actually also infect liver cells. And there's work going on to figure out what happens with that.
Yes, sir. And the next question is, will long-term supplement of substances like creatine, glutamine lead to severe effects on liver?
Long-term supplementation of creatine, glutamine, I'm not very sure about that, in the sense that it depends on what you're trying to treat. If there is chronic liver diseases where these substances are, or creatine, I'm not sure. Creatine is generally cleared by the body. Glutamine is a amino acid that is made by the liver. If those things are lower in the body, they will create some problem. I mean, if these things are accumulating in the body, they can create more significant oxidative stress and injury in the liver. And so maintaining their levels at a certain level or keeping them low is more important and critical for general health. Glutamine is made in the liver. Creatine is not so much of a liver thing, it's more of a kidney thing. But glutamine is made in the liver and is an important way of getting rid of nitrogenous waste, because what you're really doing is taking glutamic acid and putting the nitrogen on it, right? And where does the nitrogen come from? Well, it comes when amino acids are destructed or recycled, and so you have all these extra amino groups that are put on this glutamic acid and making glutamine, and that is a backup system that liver uses to get rid of nitrogenous waste. The major system, of course, is making urea, right? The urea cycle. But if urea cycle is over, then we can have these as well. And so getting rid of that is more important, I think. And yeah, it can affect the liver.
Yes, sir. And the one more question.
Is fatty liver dangerous?
Dangerous.
Yeah. So yes, it is. I mean, so as you probably already know, there is a disease now we identify, and there's some discussion now what we should call it, but it is currently called non-alcoholic fatty liver disease. During late 80s, early 90s, liver doctors and pathologists started seeing patients which had liver disease just like alcoholic liver disease. Alcohol-induced liver disease is well known. People have studied it for a long time. We know how the injury looks, etc. These patients were looking like that, but the patient themselves and their families said, no, these people don't drink. They never had a drink or very rarely drink. So the doctors were confused about what happened with these patients. One of the common factors in these patients was they were big. They're all either obese or they had overweight and so on. And then slowly now there is a realization. Now we clearly know that overnutrition, as it's called, is a major driver of non-alcoholic fatty liver disease. There is currently discussion of changing that name from non-alcoholic to metabolic or metabolism associated liver disease, but we don't know when the change of name will happen. But that's what it is, overnutrition. What happened is, as world economies got better, and this is a classic case in India, right? Since 90s, things are getting better. Growing up, I never knew anything. We never knew fast food. We never knew McDonald's or anything. Now it's very common. Everybody's going around having a burger, right? And so overnutrition, bad nutrition has become a problem, and that has led to fatty liver disease. There are two parts of that disease. One is initially you get fat accumulation in the liver, okay? And technically, from a clinical standpoint, that is not considered dangerous. So if you go to a doctor and they see a little bit of fat in your liver, they're not going to be worried about it. They're going to say, oh, you got to do some exercise, eat better, and you're okay. As the disease progresses, the fat is attacked, the liver is attacked by immune system, and you get inflammation in the liver. And that's called as non-alcoholic steatohepatitis, or NASH. When NAFLD converts into NASH, that is irreversible. And that can lead to either liver failure or liver cancer. And so that is where a lot of research is going on. Now, initially people thought, oh, inflammation is bad and that's what the issue is. Then now people are realizing that a little bit of fat that we were not worried about, we should be worried about, because that fat can produce other things in the liver that can make liver susceptible to disease. So, long answer for a short question, but yes, fat in the liver is dangerous.
Thanks a lot, sir, for answering the questions patiently. Now I call upon Ms. Preeti, Assistant Professor, to give the vote of thanks.
Thank you.
Good evening, all. On behalf of the department and its entire fraternity, I take this opportunity to propose the vote of thanks for today's session. I deem it a great honor to thank our distinguished guest, Professor Dr. Udayan Apte, for accepting and inspiring a student with this thought-provoking address. Today's webinar was full of knowledge and interesting research ideas, and it gave deep insight into the topic. Very informative, sir. I'm very much sure the precious knowledge that you gave will definitely help our students to fire up their research path. We hope to extend your cooperation for our future programs too. Your valuable presence today is a real honor to the legacy of our department. Thank you again for being with us, sir. At this moment, I'm pleasure to thank our patrons, our Honorable Chairman, sir, Dr. B. Babu Manoharan, Managing Director, Ms. Jessipriya, and Director, sir, Mr. Shashi Shekhar, our Principal, Dr. Vadi Seshagiri Rao, and our Dean Ma'am, Dr. Parvada Vardini. for permitting us to organize such webinars in spite of a pandemic crisis. And my sincere thanks to our conveners, Professor Dr. G. Srikumar, Head of the Department, the creative head behind the event, and Madam Dr. Renuka Vishwanathan, Professor, Head of the Department, Lab Affairs. I also thank my student coordinator, Mr. Alfred Francis, for his support in immaculate execution of this event. I thank my department faculty members and students for your consistent cooperation and patience listening till this moment. Hope to meet you all in the future events soon.
Thank you.
Thank you all. Have a nice day. Over to Alfred.
I once again thank all the participants for attending the webinar, and I would like to inform everyone that there will be more BioTalk series in the upcoming weeks, and it will be communicated shortly. Hope to see you all soon. Thank you and stay safe.
Right.